So when some allergen enters inside the body, then these basophils gets active, right? They release the histamine, so vasodilator increases the, it is also known as the inflammatory substance, right? Actually it increases the diameter of the blood vessels and slows down the flow of the blood, right? And at during this time duration, WBCs and some other soldiers or the macrophage cells destroys that particular allergen, right? And again serotonin, which is known as the vasoconstrictor, it gets normals the diameter of the vessel, right? And if there is any kind of chemical comes with the allergen in our body which coagulates the blood, right, which coagulates the blood, so heparin prevent from that clear clotting. So this is about the basophils. If the amount of WBCs is increased abnormally, right, if amount is increased abnormally, then normal count, clear? So what will happen, that condition is known as the leukocytosis, or which is also known as, mainly it is called the leukemia, right, that is the blood cancer. And if the amount of RBC is less than the normal count, then it is called the leukopenia, clear? So this is all about the WBCs or the leukocytes, clear? Now come to the next one, that is platelets. Which is also known as the thrombocytes, right, which is also known as the thrombocytes. Thrombocytes, as its name suggests, thrombo, that is thrombus, that is clot. That means these are the cells which helps in making the clot, or which helps to clot some chemical or some body fluid at the time of the injury, right? Within 10 minutes, the clotting time is 2 to 8 minutes actually, right? And within that time, clotting occurs just due to the presence of the platelets. Actually, they are the source of the growth factor, right? They are also the source of growth factor, clear? In NCERT, one line is marked here that they are the clear cell fragments, right? They are the clear cell fragments produced from the megakaryocytes, right? These are the platelets, these are the source of the growth factor, these are the cell fragments which is produced from the megakaryocyte. This is the question of pre-medical, clear? Now listen, actually its normal count is 1.5 to 3.5 lakh, in medical data and in NCERT, both datas are little bit different, right? So but in NCERT we have to follow the NCERT data for the NEET purpose. That's why the normal count of platelets is 1.5 lakh to 3.5 lakh, right, per cubic mm of blood. Platelet release in platelets cytoplasm, there is enormous amount of thrombostatin is present, right? Thrombostatin is found, right, and which helps in the formation of the thrombokinase enzyme, thromboplastin, thrombokinase, all these things are formed by that. Thromboplastin is formed, then thromboplastin form the thrombokinase. Thrombokinase helps in the activation of the prothrombin in presence of the calcium, right? And in see that damaged cells at the time of injury, damaged cells release thromboplastin. That thromboplastin forms the thrombokinase enzyme. It forms the thrombokinase enzyme, right? That thrombokinase enzyme, in presence of calcium ion, it activates the prothrombin. Again, in presence of calcium ion, and it activates the prothrombin into thrombin. Into thrombin, right? Now what will happen? Again, that thrombin, in presence of calcium ion, it activates the fibrinogen. It's just a short, right? It's not exact method. Actually, the blood coagulation is a big method in which we will follow two pathways, that is intrinsic pathway and extrinsic pathway, right? But it's just short summary, just to very little summary. Actually, it's not short, it's very short little, right? Just to understand all these things, and question comes to you in NEET just in that particular portion also, right? So that fibrinogen, in presence of calcium ion, active into the fibrin, and fibrin plus injured cells, right, both form the clot. So in that manner, platelets helps in the clotting. Actually, the platelets are mucilaginous surface. Pure platelets have the mucilaginous surface. So when they comes in contact with the external atmosphere at the time of injury, then what will happen? They all these stuck with each other, and some amount of blood RBCs and all these also stuck with the platelets. That's why the clotting of blood occurs, right? In some diseases, the amount of platelets get reduced, like the dengue, right? Dengue. In these platelet amount get reduced, right? So what will happen if the amount increases abnormally? If amount increases abnormally, right? Then what will happen? Amount increases abnormally, then it is called the thrombocytosis. And if it is decreased, then the normal count, then it is that condition is known as the thrombocytosis. right? That condition is known as the thrombocytopenia. Clear? So this is all about the blood cells, the conditions related with them. Clear? Now come to the blood groups. They are very, very, very important from the examination point of view. What are the blood groups? They are the genetically controlled antigens and antibodies. Blood groups are genetically controlled antigen and antibody. In blood groups, ABO blood grouping is there, in which there are majorly 4 groups: A, B, AB and O, right? These capital letters, all these shows the antigens actually. This is O, this O is, that is 0. That means antigen is absent, right? That is antigen is absent. If a person is having blood group A, for example, I just, right, as I told you, that is the antigen antibody interaction, right? Genetically controlled antigen antibody interaction. Antigens represents capital, antibodies represents small, right? Just to memorize it, right? We differentiate like that. And capital A antigen is combines with the small b antibody. Clear? Capital A antigen have the small antibody B. So what will happen? And if the patient is positive, right, so what will happen? This, it shows the antigen. This is antigen, antibody, and that positive sign is related with the Rh factor. That positive sign is related with the Rh factor, which is taken from a rhesus monkey, which is taken from the rhesus monkey. That's why rhesus Rh, it's a kind of protein actually, right? It is a kind of protein, and its gene is present on chromosome number 1. Clear? If it is present, then it is positive. If it is absent, then it is negative. And in most of the cases, positive is dominant in nature. That's why we say that the negative blood groups are very rare found, right? Negative blood groups are found to be very rare. Clear? So this is just an example to understand that what is antigen, what is antibody, and what is that Rh factor, why we use the A negative, O negative, AB positive, right? Now listen carefully. In NCERT a chart is given. Antigen is known as the agglutinogen. Antibody is known as the agglutinin. Antigens are present on the surface of RBCs, right? Present on the surface of RBCs, right? Antibodies present in blood plasma. All these are the question of pre-medical, right? Antibodies are present in the blood plasma. So now what will happen? Antigen antibody interaction, and now what is that? Listen carefully. A antigen, B antigen, A B antigen, and this is 0, that is O, right? Antigen, when we talk about the antigen, antibody, and the donors group. That means which kind of blood they received. A antigen, antigens are, oh sorry, B, AB, both antigen are present, and 0, that is antigen is absent. But when we talk about the antibody, antibody B, antigen A, antigen B, antibody A, right? In this, antibodies are absent, and in 0, that is in which antigen is absent, both antibodies are found, right? Antibody A and B both are present in the O blood group, clear? Because antigen is absent here, clear? Now what will happen? The donors group, when we talk about the donors group, that means if the A blood group people need the blood transfusion, then what will happen? What kind of blood they receive? Then that A received from A and O, right? B received from B and O. That AB received from anyone, AB, A, B and O. Why? Because in that antigen is absent, right? And in both antigen is present is here, so there is no foreign particle, and O is received from only O, clear? On that basis, the question asked is about the universal donor. So memorize that universal donor is O negative, right? Not O positive, because O positive can donate their blood only to positive blood groups, right? But O negative, because absence of the Rh factor, right? That O negative can donate their blood to anyone, right? If it is positive or negative, it's no there is no dangerous, or there is no danger in that case, right? That's why the universal donor is O negative. Universal acceptor or universal receiver or universal recipient, actually, right? AB, clear? AB can receive the blood from any blood group, clear? So this is all about. Now what will happen if by mistake some person blood transfusion occurs, and they give the A blood group to B, then what will happen? Due to antigen antibody interaction, due to antigen antibody interaction, RBCs clump, right? There is clumping of RBCs, there is clumping of RBCs, right? And clumping of RBCs is known as the agglutination, right? Clumping of RBCs is known as the agglutination. Clumping of RBCs is called the agglutination, clear? Right? So in that's why we cannot be able to give the incompatible blood groups, clear? or we are not able to transfuse the incompatible blood groups between the incompatible blood groups. Now what will happen if there is incompatibility during pregnancy, right, and due to which the hemolytic disease of newborn baby will be occur, right, and there is one more condition, that is erythroblastosis fetalis. Clear? What will happen? During incompatibility during pregnancy, if mother blood group is A, right, and fetal blood group is B, then what will happen? Sometimes mother's plasma attacks on the fetal RBC, and in mother's plasma antibody is B present which attacks on RBC. So what will happen? Interaction, due to that interaction they destroy some fetal RBCs, and due to the destruction of fetal RBCs, right, what will happen? Infantile jaundice. Clear? Infantile jaundice is there, right? That's why we say that due to that hemolytic disease of newborn baby occurs. Clear? One more condition is here, that is erythroblastosis fetalis, due to which also hemolytic disease of newborn occur. Erythroblastosis fetalis. Clear? What will happen in that case? If mother is Rh negative, right, and fetus is Rh positive, listen carefully. Dominating nature, Rh positive shows the dominating nature, but it doesn't mean that it always Rh positive, right? Because its gene is present on chromosome number 1, that is autosome, right? So it comes from anywhere, from mother or father. No, we cannot say that only the father is responsible for that, or only the mother is responsible for that, right? But we can say the Rh positive shows more dominancy than the Rh negative, but it doesn't mean that ki Rh negative is not inherited in next generation. Clear? So what will happen? In first pregnancy there is no complication, right? In first pregnancy there is no complication. First baby escape the complication of birth, right? But what will happen at the time of surveillance of the umbilical cord, right, when we cut the umbilical cord after the clumping, right, at that time little bit amount of blood of the fetus get mixed in the mother's blood, and mother's body don't know about the Rh factor, right? So it is the foreign particle for the mother's body. So what will happen? Antibodies within 1 month of after the postpartum period, that antibodies is formed against the Rh positive in the mother's body, right? And when that female conceive the second child, and again then that second child is Rh positive, then what will happen? For mother's body it is foreign substance, right? So foreign tissue or harmful. So what will happen? The antibody which is formed against that Rh positive, they destroy that fetal, Clear? They destroy that particular tissue, they attacks on that, and that's why sometimes the miscarriages is occur, right? Miscarriages will be possible. And what will happen to avoid these consequences when we test the childbirth blood group just after the birth and all these things, right? And if is there any situation like this, then doctors prescribe 3 medicines to the females, which is inserted in the females, that is, which acts against the, which destroy the Rh positive antibodies in the females, that is Rogam, Rolin, and anti-D, right? So this is all about the blood group and the antigen antibody interactions. Clear? Now come to the lymph. Lymph is the colorless fluid, right? And in lymph, colorless fluid, clear? RBCs and platelets are absent. RBCs and platelets are absent, only WBCs are present in the lymph, right? Lymph is formed by the filtration of blood, right? How? When to maintain the osmotic gradients or to maintain the osmotic pressure, right? What will happen when from blood vessel the some amount of fluid is filters in the interstitial spaces, and lymph capillaries absorb these interstitial fluid, right? And what will happen? These fluid is go through the lymph nodes and all these things, then when they flow in the lymph vessels, it is called the lymph, which is colorless fluid, right? When that lymph enters in the lymph nodes, right? What will happen when they enters in the lymph nodes? When they enters in the lymph nodes, then here by the absorption of the water and some nutrients, there is the lymph gets concentrated, and when bacteria or the microorganisms comes in contact with the lymphocytes, so WBCs attacks on it and destroyed them, right? So in that manner lymph acts, and after that lymph is again drained into the blood vessel, right? That lymph is drained into the blood vessel, clear? So in that manner that lymph is acts, clear? So this is the colorless fluid which helps to make our immune system strong, right? Now come to the circulatory system. There are 2 types of circulatory system. First one is the open, second is the closed. Open does not remain confined into the blood vessel. Right? Closed blood remains confined into the blood vessels, right? In open circulatory system, in open circulatory system, it flows in the empty spaces, right? That blood is flows in empty spaces, which is called the sinusoids. Which is called the sinusoids or the sinuses, right? Which is called the sinusoids or the sinuses, or we can say it is known as the hemocoel, right? It is known as the hemocoel. And the fluid which is filled in that hemocoel, specifically in case of insects, right? This is called the hemolymph, which is the colorless fluid, right? When we talk about the closed circulatory system, when we talk about the closed circulatory system, we know that the blood remains confined into the blood vessels, right? And for examples are more important for closed and open both, right? In open, examples are the arthropods, and some molluscans, right? In case of closed circulatory system, some annelids. Closed circulatory system starts from the Annelida phylum, right? Some annelids, some molluscans, such as the giant squid, and all the vertebrates, right? This closed circulatory system, the father of closed circulatory system is known as the William Harvey, right? The father of closed circulatory system is the William Harvey. Actually, the closed circulatory system is divided into two parts. First one is the single circuit circulation, second one is the double circuit circulation, right? Closed circulatory system, two types. First, single circuit circulation, right? Single circuit, why it is called single? Because the blood passes through the heart just single, one time, right? For example, it is present in the fishes. It is present in the fishes, and fish's heart is known as the venous heart. Why it is called the venous heart? Because fish's heart always carries the deoxygenated blood, right? When the blood goes through, that deoxygenated blood goes towards the gills, what will happen? Oxygenation occur, and that oxygenated blood directly enters into the tissue, right? That oxygenated blood directly enters into the tissue. So fish's heart carries only deoxygenated blood, that's why it is called the venous heart, right? Fish's heart is two chambered heart, right? Fish's heart is two chambered heart. Clear? Fish's heart is two-chambered heart, right?